this video, we are going to see the organization of genes. How the genes are going to be organized into chromosomes. That means a DNA is converted to or organized to genes. These genes are organized to chromosomes. How it goes into chromosomes is what we are going to see. So, at first we should know what is chromatins. Chromatins are the compacted organization of the DNA. The compacted organization of a DNA is known as chromatins. They are found to be inactive. In general, the chromosomes are found to be inactive, but a DNA is the active form. The inactive form of chromosomes are very important as they must be inert when there is no replications or cell division to be taken place. When there is no cell division, the DNA must be inactive. The DNA is converted to a compacted form of chromosomes and became inactive. Therefore, here we have mentioned that structurally inaccessible and functionally inactive. When it forms chromatin and chromosomes, this structure is not to be used for the functional actions like cell division. Therefore, they are inactive. Within the cell, the DNA is going to be associated with the proteins and get a compacted form. This compacted form is known as chromosomes. The DNA compacted form is known as chromatin. When the DNA compacted form of chromatin is with proteins, the proteins and chromatins together known as chromosomes. These are the inactive form of DNA. This organization of DNA into a chromosomes are very important in both prokaryotes and eukaryotic cells. For the organization of a DNA, it is very important because to fit the 2-meter length of DNA into a 10 to 15 the diameter of the nucleus. Here we can see that nucleus is going to be 10 to 15 meter only. But here a single DNA can have a length of 2 meter. So here the chromosomes are found to be inside the nucleus. Because of the DNA, 2-meter length DNA, not a single DNA, like that a multiple DNAs are going to be packed into the chromatin, and the chromatin with the proteins compacted form is known as chromosomes. This compacted form can fit into the nucleus. Therefore, the lengthy DNA can fit into the small nucleus of 10 to 15 length of diameter by this compaction. Therefore, the compaction into chromosomes are very important to fit the DNAs into the nucleus. Next, we are going to see what are the advantages for the organization of DNA into a chromosome. The packaging of a DNA into a chromosome is very important because a chromosome is going to the compacted form of the DNA. To fit into the nucleus or a cell, even a cell is going to be lesser in diameter than the DNA, because DNA is going to be 2-meter length, but cell is going to be lesser in length than the DNA. Therefore, the compaction of a DNA is very important to fit into the cell and into the nucleus. Second is going to be packaging of a DNA. Packaging of DNA is very important because it is a packaging of DNA into a chromosome chromosome is what makes a DNA to be inert and inactive. The inactive form of DNA is very important for the stability of a DNA into the chromosomes. Therefore, it protects the DNA from the DNA damage. The DNA damage is not taken place because of the inactive form of DNA. If the DNA is made without compaction, it can become unstable inside the cell. Therefore, the chromosomal DNA is found to be very stable form. Next, the stability of a DNA is very important. Why? It's a main question. The stability of a DNA is very important because the DNA stability causes it to be inactive form and stable form. When the DNA is stable, it can be used for next cell division to make a daughter DNA and to inherit the characteristics. Therefore, a packaging of a DNA to transmit into the daughter cells is very important. So that the stability is important. The stability can be made by chromosomes. For the next set of daughter cells, for each set of daughter cells, the stability is very important, which can be helpful for the cell division. Otherwise, when it is unstable, it gets disintegrated. When it gets disintegrated, the DNA gets disintegrated, it leads to loss of certain characteristics. But when it is stable, it can be, there is no disintegration, no loss of characteristics. Therefore, cell division occurs and the characteristics are passed to the next daughter DNA. Here, we also said that the overall organization of a DNA is very important for the gene expression. As we said that, for the expression of a gene, for the transmission of the characteristics from one generation to another generation, this is very important. The stability of a DNA is very important. So, here we can say that a main advantage of it is to pack the DNA into the smaller length of cell and nucleus and to make the DNA a stable form when there is no action of mitosis, cell division to be occurs. A stable form is very important to transmit the characteristics. So this is very important uses for the compaction of the DNA. So when we lengthen the DNA, a compacted form of chromosome is lengthened off. The DNA is very lengthy as it can, so long as it can stretch from the Earth to the Pluto 17 times. Like that lengthy DNA is going to be compacted off into a chromosome of our cell. So this compaction is very important for the stability and also for packaging of a DNA into the cell. Now we are going to see the elements for the compaction of the chromosome. Elements for the compaction of a chromosome. The first element is going to be DNA. Next, it's going to be our proteins. A DNA only compacted off, it is known as chromatin. This chromatin is compacted into a stable form by these proteins. These proteins are going to make it into a chromosome. So the chromatin is converted into a stable compacted form of chromosome by these proteins. So we are going to see about the proteins, which is a main element for the chromosome compaction. So in general, these proteins are known as histone and non-histone proteins. These histones and non-histones proteins are going to be a main element for the compaction into a chromosome. In general, half of the molecular mass of eukaryotic chromosomes are made up of proteins, and especially Eventually, these histone proteins are going to be increased amount in the eukaryotic chromosomes. As we said, there are two types of chromosomal proteins. These are elements of chromosome. First, we are going to see about the histone proteins. It is very important for the compaction of the chromosome, compaction of the DNA into a chromosome. Only by the histone proteins this compaction is going to be taken place. The histone proteins are going to be basic in nature, but a DNA is going to be negatively charged, which means the DNA is going to be acidic in nature. Due to its presence of phosphate link, phosphate is going to be negatively charged, it makes us acidic. So DNA is going to be acidic in nature, but the histone proteins are going to be basic in nature. So the histone proteins is going to have high amount of positively charged. This is a principle for this compaction to be occurs. The positively charged histone proteins can attach to the negatively charged DNA and help it for the compaction of DNA into a chromosome. So, what makes the basic? As we said that DNA has a phosphate group. This phosphate group makes it to be acidic. Like that, the histone proteins are found to be positively charged, which is basic proteins, because of its amino acids which are basic in nature. A basic amino acids like arginine and lysine are the main elements for the histone proteins. This makes them as basic in nature. As arginine and lysine are going to have a amide NH2 group in its side chain, which makes it to be a basic. Therefore, this basic amino acids can bond with the DNA which is negatively charged. This makes them for the compaction. We can see the compaction, how this linkage make them for compaction in further. Histone proteins are going to be organized into an octamer structure. As we said that histone proteins are basic in nature, this makes them to attach to the acidic DNA. But at first, histone proteins individually cannot be attached to the DNA. These histone proteins are going to form an octamer. These octamers are going to attach to the DNA. Therefore, how does this octamer are going to be formed? We are going to see that. Histone proteins has different types: H1, H2A, H1, H2A, H2B, H3 and H4. These are the 5 types of histone proteins. Among this, the H2A and H2B are going to form the top and bottom, as histone octamers are going to be disc-shaped. Here, H2A and H2B are going to be present, and here also a H2A and H2B are going to be present. At the middle, central region, core region are going to be made up of H3 and H4. So, as twice dimers, 2 types of H3 and H4 this side and that side, like H3 and H4. So, the central region of octamer are going to consist of H3 and H4. The top and bottom as a dimer, and top and bottom are going to be consists of octamer of H2A and H2B. There is also a protein H1 going to be present. It is going to be a linker protein, which is not involved in the octamer formation. A disc-shaped octamer formation are going to form a core protein. Has H2A, H2B, H3 and H4 are going to be a core protein. This core protein are going to form a octamer disc-shaped structure. In octamer structure itself, a central and top and bottom division is going to be taken place. In central H3 and H4, top and bottom is going to be H2A and H2B. Here we can see that they are going to assemble as a disc-shaped octamer. Among this, the H3 and H4 as a dimer, as a tetramer, are going to be in the central, and flank means at the top and bottom. Top and bottom is going to be covered by the H2A and H2B as a two dimers. So here we can say that H2A, H2B, H3 and H4 are going to form an octamer disc. They are the core protein, also known as core histones, where H1 is going to be a linker histone, which is not involved in octamer formation. That means core proteins are not going to be formed. These octamer formations are very involved for the first level of organization of the gene, but this H1 proteins or linker histones is going to be involved in second level of organization of the gene. The main function of this histone proteins is going to be chromatin formation into a chromosome. It is going to determine the structure of the chromosomes. It also involved in repression activity of a gene, which means inactivation of a gene, because this DNA is going to form the chromosomes. These chromosomes are found to be inactive form. Only by the binding of the histone proteins, it is going to form a compact form of chromosomes, which is an inactive form. Therefore, the repression activity of a gene is going to be made by these histone proteins attachment. So we can say that it is going to make a chromosome and also involved in inactivation of the gene. So here we can see the structure where it is going to be a top region. H2A and H2B must be top. H3 and H4 must be at the central. It is the overall structure from the top view. H2A and H2A and H2B is at the top. It is also flanked at the bottom also. It is the central H3, H4 at the central tetramer. This is central region. It is a top view. It is a side view. It is the linker H1 protein, which is not involved in octamer formation. In the octamer, H1 histone protein is not present. Now we are going to see about non-histone proteins. Non-histone proteins are very important among the other histone proteins, because the non-histone proteins are not involved in the chromosomal organization. It is not involved in the inactivation of the chromosome, but these non-histone proteins are involved in the activation of the chromosome, which means during cell division, its action come into effect, which means the non-histone proteins varies from 12 to 20. As we said that histone proteins of about octamer and also H1 proteins are to be present, but the non-histone proteins varies from 12 to 20 for the each. The non-histone proteins differs based on the condition of the cell. So the non-histone proteins, the examples of non-histone proteins are cytoskeletons. The cytoskeletons are found to be a non-histone proteins. As we know that cytoskeleton like actin, myosin, tubulin itself a alpha, beta form. These four cytoskeletal elements are found to be a non-histone proteins. Cytoskeletons are very important during the mitosis, as they are going to form a spindle fibers during mitosis. So here we can say that they are involved in chromosomal condensation also, but it has a small effect in chromosomal condensation, because it only involved in chromosomal condensation during the third level of organization. It has small effect, but its main effect is during the mitosis and meiosis function. During mitosis and meiosis, there will be a need of spindle fibers for the locating of cells, for the locating of DNA inside the cell. So, the spindle fiber formation is going to be formed by the cytoskeletons like actin, myosin, and mainly tubulin comes into effect. Therefore, the non-histone proteins has small effect in chromosomal condensation during the third level of organization, but the non-histone proteins has main effect in spindle fiber formation, mitosis and meiosis function. Therefore, non-histone proteins is also a very important proteins. It is not involved in octamer formation. So these are the non-histone proteins. After the compaction of DNA by histone 1, which is second level of organization, H1 histone protein is involved, while it is octamer, it is first level of organization, and the non-histone proteins are going to form a third level of organization as nuclear scaffold region. So, these are the non-histone proteins. These are linker histones, which is also known as H1 histone protein.